Hi guys, Mr. Holding, and this is a quick video to try to explain how we use DNA to code for different proteins and go from there. So basically, as we talked about the other lesson, we know that DNA is made up of what we call nucleotides, and nucleotides contain a pentose sugar, a phosphate group, and a nitrogen base. That essentially looks like this. Okay, we talked about the four being A, T, C, and G, and they pair A with T, likewise T with A, C goes with G, and G goes with C. And it's those sequence of codes that actually do the job of coding for particular amino acids. When we look at a DNA molecule and we look at the bases on the end of these nucleotides, we start to read a code, and we read them in threes. Okay, two ways we read them. We refer to them either a triplet, okay, or a code. In some ways, in some places we call them a codon. And essentially, this triplet here codes for an amino acid. Okay, so the DNA forms triplets. The triplets form and code for an amino acid, and then the sequence of amino acids code for a particular a protein. Okay, so each of these triplets have a different variation of T, A, C, and G, and they code for different things. So TAC, for example, is a start codon, and that means start reading, start the production of amino acids for a particular protein. And then you'll have a series of different other triplets in it that code for different amino acids. So for example, as you can see here, the triplet GAT codes for amino acid spartan, right? And then there'll be a series more, and at the end there'll be a stop code. Okay, so when we're in the nucleus, the DNA is lined out. The way that we code for this is we first of all have to unwrap it. So the DNA is in that double helix. We have to unwrap it, which exposes one of the strands. Okay, what happens then is the mRNA comes in and binds onto it, creating essentially an opposite coding. So same rules apply for the base pairing rules. It matches up with it, and that's called messenger RNA. That messenger RNA then essentially copies the code and then exits the cell. As you can see down here in this diagram here, we've got look what the messenger RNA here. Okay, this process here is called transcription or transcribing. It's another word for writing down, but essentially it's a making a copy of this here. Even though it's not exact, it's the opposite. It allows it to make a copy of it, and this mRNA exits this. The next step along this process is called move myself out of the way here, is translation. And translation is where we start to read. We translate the code, we read it. Okay, so we start to look over here. This is the messenger RNA, AUG, that gets read, and that's methionine. That's a starting codon. Okay, UAC is a codon for tyrosine, and GGU is a codon or a triplet for glycine. And those are amino acids that combine to produce a particular protein. Okay, if you think about bodybuilders, they take amino acids to build protein, protein to build muscle mass. All right, that's why they do it, because they're looking for the amino acids. And this is the same sort of thing. We're coding for amino acids. Okay, so the mRNA codes for amino acids, the amino acids join together to form proteins. Okay, so that was a very quick introduction to coding. So as we know, the DNA forms triplets in those series of threes. Those series of three code for a particular amino acid. Those amino acids codes for a protein. We have a start codon which starts it and a stop codon which stops it. All right, and the process involved in that are transcription, which is the reading of it, of the code, and translation, which is converting it from the code into a particular amino acid and then therefore a protein. All right, just going to put this video on. It'll go it's a little bit longer, 4 minutes. It's worth watching the extra time because it goes through a little bit more detail than I've just given. Okay, I hope you found this helpful.
DNA is very long and cumbersome. The strands of DNA also code for many different proteins. To get the code for the desired protein from the nucleus to the ribosomes in the cytoplasm, a copy of the small necessary section of DNA will be made. The copying process is called transcription, and it occurs in the nucleus. It's the first step in protein synthesis, the process of making protein. The second step is called translation. A portion of the DNA is unzipped so that the mRNA can be made from the DNA like a template. That's transcription. Nucleotides of RNA match with one strand of DNA and make mRNA. RNA polymerase unzips the DNA and puts RNA nucleotides into the right place. C and G nucleotides match up. A and T nucleotides match up, but anytime the RNA places a complement to an A on the DNA, it places a U instead of a T in the RNA. Remember, RNA uses uracil instead of thymine. So, if the DNA strand looked like this, the complementary mRNA strand would be the complement, but each time there would normally be a thymine, there's a uracil in its place. The mRNA also doesn't copy the entire strand of DNA. It only copies the portion of DNA that codes for the protein it wants at the time. In fact, most of DNA doesn't code for anything. Only about 2% of it codes for protein. So this small section of coding DNA has a start and stop signal. After the mRNA is formed, it moves out of the nucleus through a nuclear pore and goes into the cytoplasm. Remember, there are 3 different types of RNA. The purpose of the mRNA is to get the protein code out of the nucleus without pulling the DNA out of the nucleus. Then the mRNA will need to find a ribosome in the cytoplasm. Ribosomes can be found on the rough ER or floating in the cytosol. mRNA is read 3 bases at a time, and these 3 bases are called codons. Now, the ribosomes are made of protein and RNA, and the ribosomes are the protein-making machines that read the mRNA code and add the correct amino acid using tRNA. Remember, tRNA stands for transfer RNA, and that's because its purpose is to transfer the right amino acid to the ribosome to build the protein. The tRNA has an anticodon on one end, which will match a specific codon on the mRNA, and has a specific amino acid on the other end. Together, these 3 parts will make translation happen. Translation occurs in the cytoplasm of the cell wherever there's a ribosome. To get translation started, mRNA attaches to a ribosome and a start codon must be read. It's usually AUG, and I remember that school starts in August, and that helps me remember AUG. The first amino acid is brought in by tRNA. The anticodon on tRNA matches up to the codon on mRNA. Then the next tRNA molecule moves in and matches up with the mRNA codon. This time, the amino acids form a peptide bond and link together. Then, the first tRNA can detach, and the mRNA shifts through like ticker tape, and the next tRNA molecule can come in. The protein grows until a stop codon is reached. Once the stop codon is read, the protein is formed and ready to finish folding to become functional. And that's the end of protein synthesis. Now, we can figure out the sequence of amino acids using the mRNA and amino acid chart like this one. Let's use an example. Starting with the first 3 letters, AUG, we can find the amino acids. The first letter is A, which narrows it down to this row. The second letter is U, which narrows it down to this column, and the third letter is G, which means that this is methionine, which is the starting amino acid. Next is CCC, which is the second row, second column, and second row on the right. But we don't even have to look at the third letter in this case, because every third letter will bring the same amino acid, proline. Next is GGC, fourth row, fourth column, second row on the right. That gives us glycine. And lastly is UAA, row 1, column 3, row 3, and that gives us stop. There's no amino acid here. It just means that the protein will detach, and it marks the end of protein synthesis. Thanks for watching this episode of Teacher's Pet. Don't forget to like and subscribe, and